The overall goal of this procedure is to apply advanced fluorescence confocal microscopy to monitor spatiotemporal dynamics of signaling events during chemosensing in Dictyostelium Discoideum cells. First, chemotactic competent Dictyostelium Discoideum cells are prepared. Next, a controlled chemoattractant gradient is imaged to establish a linear relationship between the chemoattractant concentration and the fluorescence intensity chemoattractant. The third step is to obtain an immobile cell system for imaging signaling events involved in cyclic AMP gradient sensing. The final step is to simultaneously monitor two GPCR-controlled signaling events in live cells, heterotrimeric G protein activation and PIP3 production. Ultimately, spatiotemporal changes in fluorescence intensities in single cells that are simulated with a chemoattractant, cyclic AMP, can be visualized through live cell imaging with advanced confocal microscopy.
The main advantage of this technique is to allow us to obtain spatial temporal information on signaling event in live cells. These live cell measurements permit a better understanding into how a GPCR signaling network is able to detect a chemoattractant gradient and to generate proper responses. Demonstrating the procedure will be Dr. Xuehua Xu, who is an expert in live cell imaging methods.
To generate D-discoideum cells that are chemotactic to the chemoattractant cyclic AMP, harvest cells growing in D3T-rich media from a shaking culture at 22 degrees Celsius. Wash cells twice in non-nutrient developmental buffer.